Today, we're diving into some absolutely landmark research that might have just solved a puzzle at the heart of Huntington's disease that has stumped scientists for decades.
So, let's set the stage. Huntington's disease is a tragic story, and it's one that's written in our very genes. From the moment someone is conceived, the blueprint for this awful neurodegenerative disorder is already there, encoded right into their DNA. It's a lifelong condition.
But here's the thing that's driven scientists crazy for generations. I mean, if the genetic flaw is there from day one, why do people get to live healthy, totally normal lives for 40, maybe 50 years before the disease just wakes up? What's the trigger? What's going on in that long silence?
To really get why this new breakthrough is such a game changer, we first have to appreciate the puzzle that researchers were up against. And it really all boils down to three core nagging questions.
Okay, so first, there's that long wait we just talked about. Decades of nothing. Why? Second, why is Huntington so picky? It goes after one very specific type of brain cell, this thing called a striatal projection neuron, and pretty much destroys it, while leaving its neighbors mostly okay. How does it do that? And third, what is the actual toxic trigger? I mean, how does a gene that you've had your entire life suddenly flip a switch and turn into a killer?
Well, it turns out the answer seems to lie in a completely radical new idea. That gene isn't just sitting there, it's changing. It's dynamic. It's almost like a ticking clock hidden away inside the DNA of our brain cells.
The key idea here is called somatic expansion. Now that sounds super technical, right? But the concept is actually pretty simple. That faulty bit of DNA, it's a repeating sequence of genetic letters, CAG. It doesn't just stay the same size. Nope. Inside the very neurons that are the most vulnerable, that CAG repeat actually gets longer and longer and longer as a person gets older.
You can kind of see it here. A person might be born with a gene that has, let's say, 40 of these CAG repeats. But over the years, inside those specific brain cells, this expansion process starts up. It's slow at first, but then it starts to pick up speed, stretching that sequence out more and more.
And the scale of this change is, well, it's just staggering. Researchers were able to measure this at the single-cell level, and what they found was wild. A repeat that started at a length of about 40 when you were born could grow to be over 100, 200, even more than 500 repeats long in some of those brain cells.
So, if the gene is constantly getting longer, that leads to the most important question of all, doesn't it? At what point does it actually go from being just a faulty gene to being a toxic one? Well, it turns out the answer is incredibly specific.
Yeah, think of it like a fuse on a bomb. It's burning, it's getting longer and longer, but when does the bomb actually go off? When does the neuron start to get sick and die?
This, right here, is the absolute core finding of the whole study. Get this. Below 150 CAG repeats, even after decades of this expansion, the gene seems to be harmless. The researchers couldn't find any detectable toxic effects inside the cell. But the second it crosses that 150 repeat line, boom, everything changes. The gene becomes profoundly toxic, setting off this cascade of massive changes in how the neuron operates.
This number, 150, this is the key. It's the tipping point. It is the switch that flips a seemingly benign gene into a cellular assassin. And just to be clear, we're not talking about the gene length you're born with. We're talking about the length it reaches inside the cell after decades of that ticking clock.
So once they figured out this tipping point, the scientists could finally build a complete model of how a healthy neuron slowly travels down this road to self-destruction. Let's walk through the process they mapped out.
The researchers themselves describe it as a series of phases, a journey that's driven entirely from within the neuron by its own expanding gene. They gave it a name, too, the ELongATE model.
Okay, so it all starts with phase A, what they call the slow tick. This can last for like 50 years as that repeat just inches its way up from 40 to about 80. Then comes phase B, the fast tick, where the expansion speeds up big time, rushing towards that 150 repeat threshold. Now what's crucial here is that in both of these phases, the neuron is still healthy, but once it crosses 150, it enters phase C, an identity crisis. It literally starts to forget it's a neuron. Then in phase D, it hits self-destruct mode, turning on genes that basically tell it to die. And finally, phase E, the neuron is gone.
So, with this incredible new model, we can finally go back to those three original puzzles we started with and see how this ticking clock answers every single one of them.
So why the long wait? Because neurons spend decades in that slow ticking phase A, harmlessly building up those repeats toward the threshold. Why does it target specific cells? Because those particular neurons, the striatal projection neurons, just happen to have the highest rates of this expansion. Their clocks simply tick faster. And the toxic trigger? It's not the gene you're born with. The trigger is the expansion, but only after it crosses that critical 150 repeat line.
So the most important point, the one that really just changes everything, is this. According to the scientists' models, the average at-risk neuron spends more than 96% of its entire life with a version of the Huntington's gene that is, for all practical purposes, harmless. Its only dangerous feature is that it's unstable.
And this leaves us with a final, and honestly, an incredibly hopeful thought. You see, the old way of thinking was trying to treat a disease that was already burning down the house. But if Huntington's is really a disease of DNA expansion, a ticking clock, what if we could develop therapies that just slow the tick? Could we delay that 150 repeat tipping point for so long that the disease never even gets a chance to start? The clock is ticking, for sure, but for the very first time, we might actually know how to slow it down.